This is lecture 32. It's part 5 of 7 in anabolic signaling. We have two more after this one. So we've talked about these four categories of mTOR activation. In this lecture, we'll be getting to the negative regulation. Those are positive activators for the most part. We'll be talking about negative regulation today. But let's begin where we left off last time with a narrative review of mTOR. So the purpose, they're complicated signaling cascades, MAPK, mTOR. But the purpose is metabolic regulation because there are times when you're fed. And then there are times when you're fasted. And if you're constantly growing your proteins, you're translating, synthesizing proteins when you're fasted, you're not going to live for very long. Your cells have to be able to detect fed and fasted states, and your metabolism needs to behave accordingly. So you need to synthesize protein when it's advantageous to do that, and then you need to seize that synthesis when it would put your survival at risk. But when is it advantageous to synthesize a protein? When you need to, right? When your cells are damaged and you need to repair them. So damaged cells release mTOR activators. But then also, if a cell is being loaded in potentially injurious ways, you're probably going to need to repair it soon. So let's just go ahead and start signaling mTOR now. In both of these cases, damage and mechanical loading, it would help if we were to reinforce the tissue a bit so it doesn't get injured during the next exposure, future exposures to similar stresses. So we signal mTOR accordingly. But growing during a food shortage, right? That's not wise because mTOR activity is extremely expensive metabolically. It's metabolically expensive. And so it's best to conserve calories when you have none to eat. So in the absence of nutrition, mTOR is unresponsive to growth signals. But cells can be tricked into thinking nutrition is more abundant than it is. You don't have to eat 30 grams of protein in a sitting to promote mTOR's localization, the movement of mTOR. You can just eat that whatever amount of leucine, arginine, and methionine and lysine, in those 30 grams. Just ignore the other 16 and just eat that amount of those four amino acids, and you're going to get the same effect with rag protein. So you don't need to eat whole, complete proteins to get that effect. So you can sort of trick your cells into thinking an amount of nutrition has been received. And so this makes mTOR a two-key system. One key is nutrients. You need nutrients for localization. And so you need RAG to be GTP loaded. The other key is PI3K activation, hormonal, chemical, mechanical stimulators of growth. These keep REB GTP loaded. So you need RAG and REB both GTP loaded. If you're missing one of those GTPs, it's not on. So that's what makes this a two-key system. Um, so having an abundant diet tells your cells it's okay to grow, and then getting those growth factors binding, that tells your cells that it's important to grow. Okay, moving on, we're going to talk about mTOR inhibition by AMPK. And mTOR and AMPK are both enzymes. And lots of enzymes influence athletic performance. And many of those lots do so through mTOR. But these are crude, imprecise, introductory diagrams, but they illustrate a point with like no precision. And that point is when you do aerobic training, enzymes change. They change enormously. Mitochondrial enzymes, glycolytic enzymes, those involved in lipolysis and beta oxidation, tons of others, tons of enzymes undergo change. But it's not just an increase in enzymes related to performance of endurance activity. You also see an inhibition of anabolic enzymes, inhibition of mTOR mostly. And so that's what this lecture is about, inhibition of anabolic cell signaling. And the major inhibitor is AMPK, adenosine monophosphate activated protein kinase, AMPK, AMP activated protein kinase. Um, and so AMPK helps regulate the energy status of your muscles during activity, during exercise, whatever it is. If you're distance running or playing soccer or any other, you're cycling, doing any other endurance sport, AMPK is going to be a really important regulator of your metabolism in those conditions. Um, so, in addition to helping with substrate availability and consumption and burning of substrates, it also phosphorylates Raptor, which inhibits it. It inhibits Raptor, and that prevents mTOR, complex 1, from phosphorylating its downstream targets, P70S6K and 4EBP1. And AMPK phosphorylates tuberin in a couple of spots, and that activates it. That activates tuberin. So Reb loses its GTP. Without that GTP, Reb can't activate mTOR. And then also, AMPK seems to phosphorylate mTOR directly, impairing mTOR signaling. And it's the like limited, data on this one. The major sites of inhibition by AMPK, or at least those that are best characterized in the literature, are tuberin and Raptor. This is just an explanation of its role as a metabolic regulator. It's a nice passage explaining how it works, how AMPK inhibits energy consuming pathways and promotes energy producing pathways. I don't think anyone's going to get the visual reference here, but you can think of AMPK like Saint Peter, like a cartoon version. I don't think it's like a biblical version of Saint Peter, but like a Gary Larson Far Side comic kind of caricature of Saint Peter. He's standing at the pearly gates and determining the fate of everyone in line. Like, you go to heaven, you go to hell, heaven, hell, and so on. There's no purgatory here. Pick one. I'm sending you to a destination. AMPK is like a Saint Peter in that it makes that final decision. For anabolism or catabolism, you're on, you're off, you're on, you're off. You consume energy, so you're off. You produce energy, so you're on. Right? The AMPK is really it has like divine authority over your metabolism, whether you're anabolic, whether you're catabolic. If AMPK is on, you are catabolic. But under normal conditions, you're just sitting there listening, watching this. You don't have much AMP at all, really, unless you're like on the treadmill as you're watching this. So AMPK is inactive because you just don't have that much at rest. But how do we turn it on? Um, Start by building up a bunch of ADP, okay, adenosine diphosphate. Chop one phosphate off of ATP. That process is called ATP hydrolysis. You use a hydro to lyse it, a water molecule to chop off one of the three phosphates, leaving two behind, right? So you have ADP. Um, Remember, every single power stroke of a myosin head does this. Every power stroke, every myosin head's every power stroke converts ATP into ADP. But if you keep exercising, don't do like a tiny fraction of a single contraction. Do a full exercise session and you'll accumulate a bunch of ADP. You'll go through a ton of ATP hydrolysis. Um, and you'll lose more and more ATP and build up more and more ADP. So you have to start putting back together your ATP. And to do that, you can take two ADPs and make one ATP and one AMP. It's like the opposite of Robin Hood. Robin Hood like takes from the rich and gives to the poor to make two like middle classes or whatever. What adenylyl kinase is doing, this enzyme, is it like takes from the middle class and gives to some other middle class, so to make one of them poor and the other one rich. So again, this is adenylyl kinase. It's not adenylyl cyclase, right? That's the enzyme that converts ATP into cyclic AMP. Adenylyl kinase takes two ADPs and moves one of the Ps from one ADP to the other. So that way you get ATP and AMP. Now you dispose of your ADP, right? We're getting rid of our ADP and we get some of our ATP back. Um, but we're building up AMP in the process. We're restoring ATP, but we continue to deplete it through ATP hydrolysis. But we're really building up AMP this way. So AMP is really what undergoes change during exercise stress. ATP will go down some. ADP will go up some, but AMP really goes up in a relative sense. It really goes up. Um, so again, we have a big buildup of AMP. That AMP binds to the gamma subunit on AMPK, and that permits its activation. That permits AMPK's activation. In skeletal muscle, you're going to be activating it with LKB1. You see a couple of other upstream kinases in this diagram. TAC1, that's transforming growth factor beta activated kinase 1. Don't need to know it. And then the other one, that's calcium calmodulin dependent protein kinase beta. Also don't need to know it. In skeletal muscle, the primary kinase that activates AMPK is LKB1. That's the only one you need to know. And it can't activate AMPK until AMP is bound to the gamma subunit. And then glycogen, well, we're going to talk about this soon, but glycogen binds to the beta subunit. So you see a beta subunit there too. Glycogen can inhibit AMPK allosterically that way. So AMPK, AMP gets it primed for activation. AMP binds to it and that primes AMPK for activation. But then what do you do with your AMP? Right? You accumulate it, but then where does your AMP go? Um, and remember this because we're going to come back to it. You use AMP deaminase to convert it to IMP and ammonia. Remember this process. AMP deaminase. We convert AMP to IMP and ammonia. So we start eliminating our AMP, and this changes the ratio of AMP to ATP and that ratio of ADP in there as well. We start to get more ATP relative to the amount of AMP. And so there's competitive binding of ATP, ADP, and AMP on that gamma subunit. It just depends what you have a bunch of. If the ratio of ATP to ADP to AMP changes, that affects what binds there. If ATP binds, it blocks activation by LKB1. If AMP binds, it facilitates that activation by LKB1. And then if ADP binds and sits there, it's protecting AMPK from being dephosphorylated. ADP doesn't really turn it on. AMP is what does that, but ADP is important too because it prevents the dephosphorylation. And it's not just one AMP that binds. There are three sites, CBS 1, 3, and 4. But bind the first AMP and the affinity for AMP changes. So you facilitate additional binding. This is a nice summary of activation by AMP, how AMP turns on AMPK, but it also acknowledges ADP in its prevention of dephosphorylation, which means deactivation by phosphatases. Now, that's not to say that ADP isn't important. The title of this article, it's zealous, right? The title of this one is sort of exaggerating a little bit, but ADP is the dominant controller of AMPK activity dynamics and skeletal muscle during exercise. That's probably a bit of an overstatement. It's like a partisan favoritism of the middle child. Like ATP would be the eldest, AMP would be the youngest, and ATP is what deactivates. AMP is what activates. Um, but ADP is really important too. That middle child does also have a metabolic role that's important during exercise. And then getting into the other subunit, the beta subunit, that's where glycogen binds. And part of AMPK's role is to take up carbohydrates through GLUT4 translocation. That's what insulin would do. The insulin's main function. Let's get carbs into the cell. AMPK is going to do this too. So it's helpful to know, to be able to sense carbohydrate levels within the cell as a component of that cell's total energy availability. And depending on the characteristics of glycogen, the branching characteristics of the glycogen, it can be more or less inhibitory on AMPK. Um, I'll get to this soon, but AMPK brings sugar into the cell, and then it also turns on glycolysis. So whatever sugar you bring into the cell, you immediately consume it. That carbohydrate gets broken down instead of making glycogen out of it. But chronic AMPK activation does something a little different. It seems to cause more glycogen accumulation. It's probably because you just take up so much glucose. And so you create so much G6P, glucose 6 phosphate. And tons of G6P can overpower the inhibition that AMPK would otherwise exert on glycogen synthase. So chronic AMPK activity can cause you to accumulate glycogen, but in normal situations, in most kind of all-ish normal situations, AMPK is facilitating the breakdown of glycogen. So anything that produces AMP is going to activate AMPK. Uh, so like a high altitude hike, while you're fasted, right, you're going to get a bunch of AMPK activation. Anything that's generating AMP is going to activate AMPK. But let's review how AMPK gets activated, narrative form here. ATP hydrolysis happens, right? You chop up ATP, you get an ADP, adenosine diphosphate. And then through adenylate kinase, that enzyme, two ADPs become one ATP and one AMP. And so AMP levels just keep building and building up and up and up, as long as you keep exercising or do your hunger strike at altitude or whatever it is. Um, AMP binds to the gamma subunit on AMPK. And once AMPs are bound, that permits activation by AMPK, AMPK, by LKB1. So LKB1 can phosphorylate the alpha subunit on AMPK. And now AMPK is active. And then once it's active, it does tons of things. It does a lot of things. This is my sloppy drawing of everything I want you to pay attention to. It's stimulating fat catabolism and mitochondrial biogenesis, GLUT4 translocation. So that's like, let's get sugar into the cell. And then it's turning off glycogen synthase. So you don't make new complex carbs out of that sugar that gets moved into the cell, and it turns on hexokinase and phosphofructokinase. Those are both enzymes in glycolysis. So let's break down that sugar. Let's immediately break down that sugar that we just brought into the cell. Here's another look at the same stuff. At the top, you see LKB1 activating it, activating AMPK. That's how you're doing it in skeletal muscle. And then on the left, you see activation of fat catabolism. And on the right, you see the carbohydrate catabolism through activation of hexokinase and phosphofructokinase. And you see the anti-anabolism effect of blocking glycogen synthase. And then at the bottom, you see mitochondrial biogenesis. Um, PGC1 alpha, that's peroxisome proliferator activated receptor gamma coactivator 1 alpha. Obviously, you don't need to know that. Just the main things, only the stuff that I talked about. The stuff I talked about, and the only things you need to know. And then here, you can see the relationship with um, protein metabolism. So mTOR, that's on the left. AMPK phosphorylates both tuberin and raptor in a way that turns mTOR off. It activates tuberin, which encourages its inhibition of Reb, and it phosphorylates raptor in a way that prevents raptor from recruiting its downstream target. So tuberin and raptor, those are the exact same sites of interaction that MAPK has. But they're the opposite effect, right? MAPK turns on mTOR through tuberin and raptor. AMPK turns off mTOR through tuberin and raptor. And then on the right side, this is protein degradation. It's catabolism. So AMPK activates the lysosomal system and the ubiquitin proteasome system through FOXO and ULK1. You know what lysosomes are? They're organelles that perform non-selective protein degradation. The ubiquitin proteasome system is selective degradation. You have these ubiquitin tags that get affixed to unnecessary or damaged or misfolded proteins, marking them for destruction. So think of it as like kick me signs, like little kick me signs, these ubiquitin little proteins, these tags. And you put these kick me signs on other proteins. Um, and when you do that, you mark them for degradation. So if you turn on AMPK, you mark more bodies with kick me signs, and so proteolysis happens in more proteins. So through both the lysosome and the ubiquitin proteasome system, UPS, AMPK increases the amount of proteolysis, protein lysis. Cells just start to like jettison their proteins, like they're on a boat and they're just throwing shit overboard so they don't sink. So in nutrient scarcity, AMPK phosphorylates ULK1, it's like UNC, UNC 51, like autophagy activating kinase. You don't need to know that. But it phosphorylates that, and that promotes autophagy. Um, and also it goes through FOXO as well. And then when you're well fed, when your cells notice that they're well fed, mTOR phosphorylates it, obstructing the interaction between AMPK and ULK1. So mTOR turns this off, and then AMPK obviously turns it on. And here's a look at the ubiquitin proteasome system or pathway. You don't need to know anything about it other than its name. But again, ubiquitin is a little chemical tag, it's a little protein tag, and it gets added to proteins. It marks them for degradation. So it's really two processes. It's ubiquitination and then proteolysis, which is carried out by the proteasomes. Here's an even more comprehensive look at AMPK and its interactions. On the far left, you can see some exogenous activators, ICAR, metformin, resveratrol. We'll talk about those in just a second. But for the endogenous stuff in this picture, you can see the gamma and beta subunits on AMPK. Remember, gamma is where ATP, ADP, and AMP bind. Beta is where glycogen binds. LKB1, that's the primary activator of AMPK. Once AMP is bound, certainly in skeletal muscle. And so AMP has to bind before LKB1 can phosphorylate it. And then you see PI3K, PKB, mTOR signaling coming in from an IGF receptor and then going through mTOR and phosphorylating 4EBP1 and P70S6K, which is listed as S6K1 here. So you know most of this stuff. So let's talk about resveratrol first. This is a polyphenol. You get it from food like nuts and grapes and berries and red wine, right? This is everyone thinks red wine is healthy because of this, right? Because it has grapes in it. But what resveratrol does is assist in LKB1's activation of AMPK. There are different mechanisms depending on the cell type, like activation of sirtuins, uh, but all you need to know is that resveratrol helps LKB1 turn on AMPK. Then there's ICAR. This is an AMP mimetic, right? It mimics AMP. It binds to AMPK like AMP would, and it permits its phosphorylation. So it's a really effective drug for cycling and other endurance sports. You see that in New York Times article, doping cloud still looms. They're talking about ICAR. So again, effective drug for endurance sports, but it would be terrible for strength sports. So the word doping is maybe a little bit exaggerated or misplaced there. But then we have metformin. This is a major drug for type 2 diabetes. And so metformin has multiple effects, but one of them, a major effect, is the inhibition of AMP deaminase. We talked about that enzyme just a few minutes ago. I told you to remember this one because we're going to come back to it. AMP deaminase, this clears your supply of AMP. It converts it to IMP and ammonia. So if you block AMP deaminase, you block the clearance of AMP. So AMP levels build up. And when AMP levels build up, you get more AMPK activity. And then when you get more AMPK activity, you mobilize GLUT4, right? And create more GLUT4 for future mobilizations. And you turn on glycolysis to start burning the incoming carbs through both hexokinase and phosphofructokinase, PFK. So that's better than insulin in terms of like if you have type 2 diabetes, is way better than insulin would do. Um, but unlike insulin, which is anabolic, if you're taking metformin, like your sprint speed, isn't going to improve your bicep girth, your bench press. These are all going to get worse or go down. So insulin is anabolic. Metformin accomplishes insulin's primary function in a catabolic way through AMPK stimulation. So here's a look at a diagram of showing the same thing, metformin. You can see one major mechanism of action of metformin is blocking AMP deaminase. So AMP levels build up. AMP, this is a ratio here. So AMP crowds out ATP for binding sites on AMPK, on the gamma subunit. And then you get all the functions of AMPK, right, which includes glucose uptake and glucose catabolism. Let's turn on glycolysis. So that sounds like really nice effects of AMPK. Um, but why would it block mTOR activity? Well, because mTOR is super expensive. And AMPK is trying to budget your metabolism responsibly. And part of that is turning on ATP synthesis. So, let's burn fat, let's burn carbs, let's build up a bunch of ATP by burning energy substrates. But another part of that equation is shutting off ATP consumption. If something costs the body a bunch of ATP, like protein synthesis does, protein synthesis is super expensive. Those things get turned off. So anything that's promoting ATP generation gets turned on. Anything that is promoting ATP consumption gets turned off. That's its St. Peter role. Here's a nice passage about why mTOR gets shut off. Protein synthesis, this can account for as much as half of a cell's energy expenditure, a third to a half. And that's a large percentage of a cell's total budget. So when energy efficiency becomes a major focus, becomes a priority, you have to shut that down if you want to conserve ATP. But what does that mean for muscle metabolism? Here are a couple of papers out of Benoit Violet's lab that characterized mice with deficient AMPK. They get about 50% bigger. The developing musculature grows more, and they grow more in response to mechanical load. So exercise and the results are more pronounced. So this is in mice with deficient AMPK. They're going to get a lot bigger. And then opposite that, if you increase AMPK levels, activate a bunch of it, you shrink, right? You induce atrophy. So high levels of AMPK correspond to low levels of muscle. But AMPK doesn't just have roles in the periphery. While it's promoting the burning of energy substrates in the periphery, it's encouraging the ingestion of energy substrates at the same time. So consumption in the periphery and consumption in your mouth. Um, so remember the major regulatory center in the brain that we talked about, the one that controls appetite, the hypothalamus. If you activate AMPK in the hypothalamus, you get the munchies. If you deactivate it, if you deactivate AMPK in the hypothalamus, satiety results. You experience satiety. So what mechanism gives leptin, right? It's named the satiety hormone, AMPK inhibition, right? In the hypothalamus specifically. And then what's the mechanism that gives people the munchies when they do marijuana? AMPK activation in the hypothalamus. So here's a look at insulin and leptin signaling in the hypothalamus, similar effects on satiety. And these pictures are looking at the hypothalamus and the periphery. So various things will stimulate or inhibit AMPK. On the left, that green box, that shows the hypothalamus. Leptin inhibits, ghrelin promotes, low blood sugar promotes, cannabinoids, marijuana, right? That promotes. So you're encouraged to eat if you're promoting AMPK activity. And then on the right, that big brown box, that's the periphery. In the periphery, lots of stuff is going to promote AMPK, including leptin. So notice the contrary effects of leptin in the hypothalamus and the periphery. I mean, it seems counterintuitive until you think about it, and then it makes perfect sense. So leptin is released from fat cells when they're growing. And if your fat cells are growing now, there's just no reason to keep eating. So leptin can block AMPK in the hypothalamus, and that's a totally reasonable thing to do. No need to eat yourself to death. But in the periphery, it's okay to start metabolizing those calories at an elevated rate. Uh, Again, you've already grown. And so disposing of a little bit of that growth by initiating lipolysis, that's probably a good decision. And so AMPK helps do that. So it has different effects, leptin does in the periphery and in the hypothalamus. All right, so let's summarize all of these. We'll start with AMPK, and then we'll move on to mTOR and other things we've talked about. So um, turn on AMPK. If you want to do this, you're going to need AMP to bind to the gamma subunit. If you bind AMP to the gamma subunit, that permits activation by the upstream kinase, which phosphorylates the alpha subunit. And exercise is really the best way to accumulate AMP. First, ATP hydrolysis. So you're going to start building up ADP. Then the adenylate kinase reaction. That's going to take two ADPs and make one ATP and one AMP out of them. Keep on exercising, and your AMP levels will go way up, and you'll find yourself through LKB1 activating a lot of AMPK. Once AMPK is active, it phosphorylates, activating tuberin, and that inhibits Reb. Tuberin inhibits Reb. It also phosphorylates, inhibiting Raptor, so it prevents mTOR from kinasing its downstream targets. And then there's evidence that it's phosphorylating mTOR directly, inhibiting it. And the reason AMPK does this is how expensive mTOR is. Protein translation costs the cell a lot of energy. So if ATP is something we care about, if we're trying to conserve it, if we're trying to be, a conservationist here, this is a good process to shut down. And then here's a look at the map of AMPK-induced protein degradation, inhibition of mTOR, that's on the left. and promotion of the lysosomal and ubiquitin proteasome systems of degradation, that's on the right. All right, so let's summarize mTOR itself. The mTOR system, right, this is the major way that your cells create new proteins. You're going to translate new proteins, but mTOR has to be really finely tuned, tightly regulated, because you don't want to grow in times of starvation, and you do want to translate a bunch of proteins, grow in that sense when the opportunity is advantageous. So there are a lot of regulators all trying to just make sure your metabolism is fit to your environmental needs. And here's the main hub for mTOR for complex 1. mTOR itself is a kinase, right? It phosphorylates stuff. The mTOR enzyme phosphorylates stuff. Raptor, regulatory associated protein of TOR, this recruits the downstream targets. So mTOR can phosphorylate them. And then Gable, this is mostly a structural protein, but in some contexts, it does influence function. There are also inhibitory proteins, Deptor and Pras40, that are considered part of the household of core proteins. And then immediately upstream from mTOR is tuberous sclerosis complex, right? Tuberin, technically TSC2, that's tuberin. And that's a negative regulator of mTOR. It breaks down Reb's GTP, and Reb needs that GTP to bind to mTOR, activating it. And then downstream from complex 1, we have P70S6K and 4EBP1. 4EBP1, that's an inhibitor of protein translation. So when mTOR phosphorylates it, it shuts it off. So it relieves the inhibition. And then P70S6K is a kinase. And what it's phosphorylating is ribosomal protein S6, and that promotes protein translation. So mTOR is turning that on. And then there's complex 2, mTOR complex 2. The major information here to know about is the core proteins, and that PKB is among the downstream targets for complex 2. mTOR complex 2 phosphorylates PKB, activating it. So that's the point of connection between the two complexes. mTOR complex 2 activates PKB, which is AKT here, but that's PKB. And PKB inhibits tuberin, which allows Reb to activate mTOR complex 1. And here's a roster of the chemical, hormonal, mechanical, and nutritional activators of mTOR, a bunch of stuff that we talked about. And there's a list of drugs and supplements on the right. We talked about some of this, leucine, resveratrol, metformin, icar. We're going to talk about the other stuff here as we get into our practical applications. But that's it for this lecture. Here are the review questions, and I'll see you in lecture 33 when we begin our practical applications. That's going to span 2 lectures, but we're done with the basics, and that's going to serve as the mechanisms to explain our applications.